Well, thank you very much for the invitation to speak at this conference. It's slightly different from the conferences I normally speak at, but great pleasure to be here. So these are my disclaimers. And I'm going to talk to you about metagenomics. And what is metagenomics? Well, metagenomics is the process by which we detect all nucleic acids from a clinical sample for the identification of infection of unknown etiology. It's generally based on a single sample, it's unbiased, and it's all-encompassing. So just to set the scene as to why we have introduced metagenomics diagnostically, I work at a tertiary referral hospital. I'm a clinician. It's a specialist pediatric hospital with local, national, and international patients. It has 67 specialist services, including rare cancers, genetic disorders, complex medical and surgical conditions. And we have a big cohort of patients with primary immunodeficiency and bone marrow transplant. So we have a lot of infection and a lot of complicated and unexpected infection. So in the autumn of last year, we were asked to be part of a network of labs that were going to deliver a method that had been developed by Guy's and St Thomas's, which is a nanopore method. It was funded by NHS England. It was aimed to be implemented during the winter of 24-25. And the idea was to carry out rapid respiratory metagenomics in intensive care patients. The turnaround time was going to be same day and also reporting the final result the next morning, so 9 to 24 hours. And it was a unified metagenomics method for the detection of any pathogen. It was untargeted, based on untargeted ONT technology with differential cell lysis to remove human nucleic acid in respiratory specimens. And the idea was to inform patient management and pandemic preparedness. So this was stood up at our hospital by a team of people. We're very used to working with metagenomics. We've been running metagenomics for 10 years. But it was led by Robbie Hammond. And we wanted to adapt this to our particular circumstances. So in the Guy's and St Thomas's experiences, they've been working with bronchoalveolar lavage, lower respiratory tract samples, and we were more interested in including upper respiratory tract samples as well. And that meant they were much more polymicrobial, so that was the nature of the sample that we were getting. We looked at the pediatric and cardiac intensive cares and also patients who were receiving ECMO. We changed the reporting a little bit because at Great Ormond Street we're very keen to have very granular detail of what the microbial community is. So we reported any bacterium that was present at 10 reads or more, even if it was low abundance. And we also wanted to report any pathogenic organism that was present at all. And that reflects what we do microbiologically on the plate. So it was just changing it to reflect our practice. We validated all our metagenomic results against culture, and we also used PCR, and even PCRs that we normally wouldn't use, we then tested anything that was positive by metagenomics by PCR. And we processed 177 samples in 3 months. As you can see from the workflow, the initial report was issued the same day. Having received the sample at 9 o'clock in the morning, the first report went out at 4 p.m. and the final report was the next day at 9 a.m. So this is a complicated slide. You don't need to focus on the details, but just to show you that this method translated brilliantly from Guy's and St Thomas's, where it had been developed, to us at GOSH. We had very high specificity, very low false positives across the board, just like Guy's and St Thomas's. The sensitivity at the same-day testing was pretty good, about 3/4 of the bacteria and half of the viruses and fungi, just like Guy's and St Thomas's. And by the next day, we also had pretty good sensitivity. And don't forget, we were reporting against a much more stringent background gold standard than Guy's and St Thomas's. So pretty good sensitivity and specificity. The performance is shown here, and you can see in light green are those pathogens that were picked up by metagenomics that were also picked up by routine culture and PCR. In dark green were the ones that we had additional PCRs that we wouldn't normally have used, and we actually confirmed their presence. And then there were one or two specimens, there were one or two things that we missed in the bacteria and the fungi shown, and the viruses shown in red. And you can see that the nature of these, the things that were missed, were largely viruses, actually, although there were one or two other pathogens that were missed. Not a huge number, and don't forget, we're reporting against a very stringent gold standard here. Did it change clinical care? Yes, it did. In about 40% of cases, as shown in the blue and green at the top left, it's top left, yes, you can see that clinical care was changed. Most importantly, it impacted on antibiotic usage, and it changed, as you see below that, antibiotic use changed the nature of the antimicrobial agents used was changed, or we continued with antimicrobials that we'd recently changed, and in some cases we stopped antimicrobials as well. And not only did it impact clinical care, but it was highly accepted by everybody. You can see that we had a survey of nurses and doctors, and 72% felt this had a positive impact on their practice and on the patient's outcomes. So we, as I say, have been running metagenomics at Great Ormond Street for the last 10 years, and our metagenomics there is actually aimed at sterile sites, not at polymicrobial sites like respiratory samples. And the aim of the metagenomics service is to identify undetected pathogens in sterile sites. We also want to exclude pathogens because we want to be able to inform the clinical teams that they can use immunosuppressive agents without worrying that they might be making an infection worse. And then as an additional thing, we also want to provide the surveillance for pathogens of high consequence. And this service was started by myself with my PhD student, Julianne Brown, and my postdoc, Sophia Mfopoulou, who are still with me working on this service. So our GOSH metagenomics service was UKAS ISO 15189 accredited last year. We focus on tissue and sterile fluids, and those are listed there. And since 2014, we've done over 500 pediatric and adult samples. Now, because we're accredited and we're better known, we're doing about 200 to 300 samples a year, and 60% of those are not from our own patients. This is a sensitive methodology. The lower limit of detection for RNA pathogens is about 10 to 100 copies per mil, so approximating to PCR. But for DNA, although it's good in fluids, it's slightly less sensitive in tissue. We have identified a previously undetected pathogen in 25% of cases, and that rises to 33% where the patient is immunocompromised. In 75% of cases, the metagenomics result has changed management. We're now linked, in fact, to 3 multidisciplinary team meetings, one in adult encephalitis, one in pediatric encephalitis, and now more recently in ophthalmological MDT. And the advantage of this methodology is that it preserves the host RNA and DNA, so we can actually use it for host biomarker analysis. This is just to show you some of the organisms that we have discovered, which have been, and in pink are those that were found to be causing either an encephalitis or a hepatitis and had never previously been described to do this. We have identified 9 new pathogens, and in 11 cases, or actually it's now 12, we actually have found the pathogen only in tissue, only in a brain biopsy, and not in any other sample. We confirm any new pathogen that we get by doing extensive studies. We do PCR, we do immunohistochemistry, we sequence the genome from the sample. And you can see here a few of the cases where we only found this pathogen in tissue and not in the cerebrospinal fluid. And this has actually prompted a new diagnostic pathway whereby patients are now advised if infection is still in the mix, despite everything being negative, then we now proceed very frequently to brain biopsy for encephalitis and to liver biopsy if we have a hepatitis. and that is actually amazingly safe. So just quickly to compare the two respiratory and the sterile site metagenomics, our current metagenomics at GOSH is accredited. The GSTT one is now actually being submitted for accreditation. We focus on sterile sites, that one focuses on acute pneumonia. We are a diagnostic test of last resort. People will have had almost every other test that people can think of. The patient will have had that before they come to us, whereas the respiratory metagenomics is first line. Our aim is to detect and exclude infection, whereas it's faster detection of respiratory and antimicrobial resistance for the respiratory metagenomics. The turnaround is long for the Illumina method. It's at least 5 days, and it can be up to 2 weeks, whereas the Nanopore method is same day. And as I mentioned, the sensitivity is high, but it's less so for the respiratory Nanopore method. And I just want to highlight the fact that we can do the transcriptomics with our current GOSH method, whereas it's not possible with the respiratory method. So we had the idea, we thought, well, if this is going well for respiratory metagenomics, why don't we try Nanopore for our sterile sites service? So what we did was mock up clinical samples using a mixture of DNA and RNA viruses. We used viruses because they were easy to get hold of and we had good reagents for these. We diluted them from single copies up to 10 to the 4 copies, and we added human DNA and RNA to mock up a high biomass sample, which is what we often use. And we tested 3 methods: untargeted Illumina, which is our current sterile site method, untargeted nanopore sequencing, and we also added in a capture method because we hypothesized that that would be better and more sensitive. So what did we find? If you look here, the dilutions are along the x-axis, from the 1 being the highest dilutions to 4 being the lowest dilution, and the bases covered are on the y-axis. And you can see that the current method, which is in red, I don't know if I've got a laser, I don't know if it works. Okay. The current method, which is in red, was pretty good, but it didn't detect some of the DNA viruses, particularly at the lowest copy number. The Nanopore was less sensitive generally, and it didn't detect some RNA and some of the DNA viruses. And best of all was the targeted enrichment of the Illumina. That method, the targeted enrichment, was also faster than the Illumina untargeted, so we could get the result out by day 3. So by the late morning, you could get the result out rather than 5 days. However, we know that there are targeted enrichment methods for Nanopore, but they involve, the ones that are described and published, involve a conversion step where the Illumina library then undergoes ligation of the Nanopore primer binding sites to allow the Nanopore sequencing. And the problem with that ligation step is that it's lengthy, it can be up to 2 or 3 hours, it's costly, and it's labor intensive. Having said that, by the time you do it, you do get the sort of same kind of output as you have from the Illumina with the enrichment. So it's not necessarily even that much quicker. So to overcome this, Oscar Torres in my group, who's in the audience, worked out a workaround in which he uses 2 sets of primers, a 4-primer pair. And what he does is he uses the primers which have the Nanopore binding site, the Nanopore primer binding site, and which will amplify the hybridization capture library. And then you can use the Nanopore primers very quickly to generate the sequencing library. And this is we call the 4-primer post-capture PCR. And this is much quicker, as I'll show you in a minute. But what this does, if you remember, this is the slide I showed you before. Nanopore is in green, the untargeted Nanopore is in green, targeted Illumina is in blue, and the targeted Nanopore is in orange. And you can see immediately that doing the targeted Nanopore brings the sensitivity of Nanopore up to what is extremely good and would be good enough for our sterile sites, and actually better than what we're doing at the moment. And this is comparison. The red now is added in to show you the comparison with our current sterile sites Illumina method. Just to show you how this compares in terms of turnaround times, I've shown you the other three before. The bottom one here is the new turnaround time, the new protocol, and you can see that this actually reads out earlier. You can see that at 1 hour it's reading out in the early afternoon of day 2, so that's a whole sort of day earlier than the Illumina method. And at 4 hours, you can still read out in the late afternoon before you go home a second result on day 2, and then with the final result the next morning when you come back into work. So just to show you how that does in terms of sensitivity, if you look at the 1 hour readout, so this is mid-afternoon of the second day, what you're able to get is all the highest copy number pathogens and also all the medium copy number pathogens. You're missing 50% and a bit more of your lower copy number pathogens. If you then read out the result before you go home in the afternoon, the second day, then it's actually at 4 hours, then you actually get everything except the very lowest copy number. So that's pretty important because often things that are unexpected are there in high copy number and we're just not testing for them. And it's not always that we're looking for something that's present in low copy number. And by the next morning, the majority of pathogens are, even at the very lowest copy number, this is 10 copies per mil, are detected. So what does this mean in terms of how we can use this clinically? Well, we have also been working on trying to improve other aspects of the pipeline. And so what we've done is we've changed the extraction method, we've looked at the duration of hybridization, and we've done extensive analysis of whether we can reduce hybridization times. And indeed we can, and we don't have any worse outcomes. In fact, in some cases, particularly for RNA pathogens, bizarrely, the reduced hybridization times gives you a better sensitivity. And if you look at that, at the bottom, the version 2, if we incorporate these new modifications, then actually we could read out our metagenomics at the same day. It's an extended day, but actually that can come down. If we get the hybridization even lower, which we think we can do, you could probably read out that metagenomics result, which is giving you everything the same day, and that will give you everything at high and medium copy number and quite a lot of what is present at low copy number before, within a day of having received the patient sample. So we're now approaching both the sensitivity of PCR and also the rapidity of PCR. And then, of course, you have to wait till the next day, midday, the second day, to actually read out your very lowest copy numbers. So just to summarize, I hope I've shown you that rapid nanopore respiratory metagenomics is faster than standard methodology for identifying respiratory infections and has similar performance. It has proven impact on diagnosis and management. We have to see still whether that leads to outcomes, better outcomes, and that's all being analysed at the moment, and we also need to look at the cost benefit. But it is actually getting the results quicker than conventional methodologies. The pipeline was developed at GSTT, but it has translated brilliantly. The methodology has worked extremely well in our hands, and I think this can be and will be rolled out to other centres. In fact, there are going to be about 30 centres across the UK that will be now running this respiratory metagenomics. So we're glad that we were able to show that it was so easily operational. And it can be customized to reflect local needs. It's more difficult to do that if you don't have a lot of experience, but eventually I think what we can do is we can customize these pipelines to reflect what is local practice. We think that targeted nanopore, and we've shown that targeted nanopore increases the sensitivity of metagenomics to that of short read sequencing. And it enables use where detection and exclusion of pathogens is critical, so the so-called sterile sites. We cannot accept a lower sensitivity methodology for those sites, particularly in the immunocompromised, but we think we've shown that targeting nanopore will actually achieve that. It also enables, the fast version enables a simple, cheap and fast metagenomics with the potential for a same-day readout for probably everything effectively. This is the first, we've only just developed this, but there are more improvements that can come, and we think that this is actually now a realistic possibility. It preserves the host transcripts, which is incredibly important, becoming more and more important. We want to be able to actually identify biomarkers of outcome, and we also want to be able to look at pharmacogenomic markers, and it can also be used both in conjunction with the metagenomics to look for those, but it can be used for anything. It can be used for host exome sequencing, the targeted method, and for pharmacogenomic sequencing, as well as pathogen sequencing. So it's a fast method that has the potential to be very widely used. So it just remains for me to thank the people who did the work. The metagenomics team in my research team is led by Oscar Torres Montague, and Oscar's in the audience, and Sarah Buddle, together with Sophia Morfopoulou. Then we have a very good genomics sequencing facility led by Rachel Williams, who was previously my postdoc. And she, together with Lisa Forrest and Liz Marina Martin and Tony Brooks, have contributed to this. Robbie Hammond, Angelica Kopek, and Julianne Brown stood up the respiratory service. And we're grateful to the Guy's and St Thomas's people for helping us to get this underway. And then also thanks to our funders who are listed at the bottom there. Thank you very much for your attention, and I'd be very happy to answer questions.